All right, this video is 9.4 non-Mendelian genetics in a unit called Mendelian genetics. That doesn't make any sense. Well, let's find out what it's all about. Let's talk about it. Mendelian genetics, of course, remember these ideas of homozygous, heterozygous, dominant, recessive. Dominant always expresses itself, recessive doesn't express itself. These are really plain and understandable things, right? And so when Mendel was doing his experiments, this is how he basically saw these really easy, plain things to see. But it turns out that not all genetics work this way. And so we're going to look at a couple of instances where it doesn't necessarily work the way that we'd think it's going to work. And the first one of those is called incomplete dominance. Look at the word. It's dominant. It's just not completely dominant. Back here, purple is completely dominant over white. How do we know? Well, even when the white allele is present, in the heterozygous condition, the purple completely dominates it. An incomplete dominance is where you have multiple dominant alleles, or more than one dominant allele, and neither one completely dominates the other. What does that look like? Well, let's look at this example. Here you have red flowers, white flowers. Both red and white in this particular species are dominant. And so when you mix them together, what happens? Well, red doesn't completely dominate white. White doesn't completely dominate red. And so you get pink. You get a blend. And so with incomplete dominance, that's the key word, is blending of the phenotype. And notice here the Punnett square, when you mix two pinks together, what ends up happening? Well, the white and the red come back out because the pink represents the heterozygous condition. Another example of this is called codominance. Codominance, more than one dominant allele still, except for this time, think of the word co. What does co mean? Like cooperate. If you cooperate with someone, you're sharing the operation, right? And so codominance means sharing dominance. This means that both dominant alleles are expressed. So you have red and white again, and in this particular case, you have red-white, not pink. And so how do you know the difference? Well, you're not going to need to memorize different flower species or anything like that. The prompt will say something like sharing dominance, or in this case, blended, or it'll use the words incomplete dominance or codominance, and you have to then come up with the desired phenotypes or something along those lines. But notice, same kind of idea. The heterozygous in this particular case is going to be not a blend, but both alleles are going to be expressed. In humans, this is present in blood types. Blood types are interesting because it also shows this multiple allele concept, which we're going to look at pretty soon. So in blood types, A and B are both dominant, and O is recessive. And so if you have A and O together, then of course A is going to show itself, or B and O together, B is going to show itself. And then you of course have AA or BBs, which is shown there. But if you have A and B together, then what blood type do you have? AB. If it was incomplete dominance, you would have like blood type C, right? Because it would be like a blend. But instead, both A and B show themselves, and so AB. And then, of course, if you have no A's or any B's and just O's, then you have type O. Type O, oddly enough, is the most common blood type. Multiple alleles. So blood types is also an example of this, but multiple alleles is where you have one gene, say blood type or rabbit hair color, but you have more than two alleles. All right, everything that we've dealt with up to this point, there's been a dominant allele, there's been a recessive allele, right? And so, whereas the dominant always shows, the recessive does not. And so with a multiple alleles situation, you have what's called like a hierarchy of dominance. So if you look at this on the left there, you have what's typically called wild type. Wild type just means the most prevalent or most dominant of the types, and wild type in rabbits is brown. And then chinchilla, which in this case is a color of a rabbit, shockingly. So chinchilla is the most next dominant, or second place dominant. It's not as dominant as wild type, but if it's there and something else is there, then it will show, right? And then Himalayan comes after that, and then white or albino is the most recessive. And so you have this like hierarchy of dominance. Human eye color is another example of something that shows multiple alleles. And then this is different than polygenic traits. Poly, many, genic, genes. So this is multiple genes coding for one trait. Human skin color, best example of this. So you have multiple genes. Not exactly sure how many. Last I read, it was 21, but somebody's going to correct me in the comments down below. Correct me in the comments down below. How many genes code for human skin color? It's a lot. And it's lots of dominants and recessives, right? And so the way that I always teach this is I think about light switches on the wall. And if you turn on all the light switches, it's completely light. If you turn off all the light switches, it's completely dark. If you turn on light switch 3, 7, and 14, it's going to be a particular lightness in the room. If you turn on 3, 12, and 18, it's going to be another version of that. And so with polygenic traits, you have different traits expressing themselves in different combinations, and that's why there's so many different skin colors available. And so think of the main idea that I want you to get for polygenic traits is multiple genes for just one trait. And this brings us to sex-linked traits, probably the most complicated portion of non-Mendelian genetics, but it's still not that complicated. And so sex-linked traits have to do with traits that are linked on sex chromosomes. And sex chromosomes are those chromosomes that determine sex. And so in this particular case with humans, the Y chromosome is what we would call the sex-determining chromosome, because if you have a Y chromosome, you are male. If you don't, you are female. And so what does that mean? Well, females have an XX chromosome, or XX genotype with their chromosomes, and males have an XY. And so the X chromosome is necessary for life. You cannot live without an X chromosome, but you can live without a Y chromosome. Half of our population demonstrates that every day, right? And so if you think about just a normal setup, like a Punnett square setup between these two, what are the possible offsprings? Well, you have, of course, XX could combine, the XY could combine, and you have a 50-50 shot here of being male or female from any two individuals, right? And so that's the idea here. And so what if a trait is just found on the X chromosome? Well, females are going to have 2 instances of that allele, right? Whereas males are only going to have 1 instance of that allele because they only have 1 X chromosome. And so this is going to lead us to some different looking kinds of Punnett squares. And so when you come across a sex-linked problem, first of all, let me say this. The most common mistake that my students make is after I teach sex-linked traits, then they pretend that every problem becomes a sex-linked problem. It's not. It's only sex-linked problems that are sex-linked problems. How will you know that it's a sex-linked problem? It will say in the prompt, such and such is a sex-linked trait. And that should switch your brain onto doing your Punnett square that looks like this. Otherwise, they're all going to look, something like this, just simple, right, like we've done in the past. But when you read sex-linked trait, you're going to fill in the XX on one side, the XY on the other, fill in the XXXY in the middle, and then you're going to set up your alleles. And so notice this particular male, colorblindness is a sex-linked trait, whereas colorblindness is recessive and normal vision is dominant. This particular male is normal, right, because he has a dominant allele. Why doesn't he have 2 of them? Well, he only has 1 X chromosome, right? And so he has that dominant allele, whereas the female in this particular problem has 2 recessive alleles for colorblindness, and so she has colorblindness. And so where does colorblindness then come from? Well, it comes from the X chromosome. And so if you are a male and you have colorblindness, who gave it to you? Your mom, every time. Because if you're a male, who gave you your Y chromosome? Your dad is the only one that could give it to you. And so, whereas if you're female and you're colorblind, then you got 1 X from mom and 1 X from dad. And so that came from both parents. There's a couple other examples of sex-linked traits. Colorblindness is the most common. Hemophilia is another one that's a recessive disorder. Marfan syndrome is a dominant sex-linked trait. And so we will have plenty of practice in class on this so that you can tell the difference between a normal problem and a sex-linked problem. Again, the prompt will be very clear. It will let you know when you're dealing with just a normal Mendelian genetics versus a sex-linked problem.